Hello. Welcome to Monday night, as we normally do here. Hope you all are doing okay. So today we're going to do the last bit of new information for this unit, which is talking about macronutrients. So that's carbohydrates, lipids, and proteins. And we'll talk a little bit about what's special about each of them. And as you can see in the objectives on Blackboard, the main thing is that you can recognize them by their functional groups. So we've talked a little bit about functional groups in a previous module, the little parts of the molecule that cause it to do whatever it is that it does. And those will be helpful in allowing us to figure out how we can identify these different types of groups as well. All right, so before getting into the first one here, let me switch over to my, there we go. All right. So these are called the macronutrients simply because they make up most of our nutrition. So micronutrients are things like vitamins and minerals that you just need tiny, tiny amounts of. Macronutrients, macro means big, are the things you need lots and lots of. So the three broad categories are carbohydrates, that's sugars and starches. Then we have lipids. Lipids are actually a very, very big category of lots of different kinds of molecules. It's all nonpolar things. So things that are without a lot of oxygen and nitrogen and things that make for polarity that we talked about in the last unit. So just for examples, the things we talk about most when we talk about lipids and that we'll look at today are things like fats, oils, and fatty acids, but also stuff like steroids, hormones. Many hormones are lipids. Some other important things that we'll see. So I'll just say et cetera, because it's actually a pretty big category of a lot of different types of things. And then third are the proteins. All right, so those are the major classes of macronutrients. If you think of any food that you eat, chances are it is primarily made of either one, two, or three of these things. There's also things like fiber, which is not digested, so we don't really get any nutrition from that. And then, of course, the micronutrients and water is a big part of food as well. But the things that actually give us nutrition are probably going to fall into one of these three categories broadly. So because this is chemistry and we're interested in how the structures of molecules influence what they do and influence their function, we're going to look today at some of the things that are common about each of these families so that you can start to see if a molecule better fits in one category or another. All right, so we'll start with the carbohydrates. This is a link from Blackboard, so if you want to review this yourself, you are welcome to click the link on Blackboard and read through this stuff. I'm not going to read through it for you. I just want to use it for the pictures while we talk about some of the important distinctions here. All right, so here are some Lewis structures of some different monosaccharides. Now, what are monosaccharides? And that word is right here. So here we're talking about a single sugar, a simple sugar. So if you've heard about simple sugars, these are the types of molecules we're talking about. They'll have 3, 4, 5, or 6 carbon atoms. And then if you notice from the pictures here, they have pretty much each carbon in the structure, look at this big one here, glucose, has an H and an OH. And if you put H and OH together, you get water, right? H2O, an H and an OH. So that's where that word carbohydrate comes from. All of these types of molecules have a formula that's like a carbon and a water. So carbohydrate. And another way of thinking about that is to look at the structure and see that each carbon has an H and an OH attached to it. Now that's not true for every single one. At one end of the sugar, it's going to have some kind of a functional group. Sometimes it's not at the end. Sometimes it's more in the middle. And then the other end, carbon always has to have 4 bonds, so there might be an extra hydrogen there. But on average, everything has one H and one OH. So things we can know right now, sugars are going to be extremely polar. We talked about how OHs allow for that hydrogen bonding, those really strong connections between molecules. And that's true with sugars as well. If you've ever tried to cook sugar or like make candy, if you just take raw, like just white sugar and you try to heat it, it actually won't melt nicely. It'll start turning brown. It'll start caramelizing before it melts. That's because its hydrogen bonding is so strong that the molecules themselves actually start breaking down before it really melts. In order to melt sugar, you have to add a little bit of water to allow it to dissociate and dissolve and break up those hydrogen bonds at a little bit lower temperature before it starts doing that decomposition. So lots of hydrogen bonding there in these monosaccharides. Now, monosaccharides have all different names based on the position of the H and the OH. So there are many, many, many different ones, and we don't need to see a whole table of them. You can look them up. Lots of different names. But if we just look at 2 of them, for example, glucose and galactose, they look almost the same. Same number of carbons, same number of oxygens, same number of hydrogens. They're isomers. They're all C6H12O6. They all have this aldehyde functional group here. The only thing that's different is the position of the H relative to the OH, so one side or the other. And we said back when we were talking about Lewis structures that single bonds rotate, and so it doesn't actually matter what angle you put them in. Well, as it turns out, when you get enough different stuff on these atoms like this, it actually ends up being an issue. So if you were to actually take a model kit, if you have access to one or a virtual one, might be a good idea for one of your at-home experiments if you want. One thing you can do is to build a model of this and a model of this, and you can twist them around and try to put them next to each other, and you should be able to see that they, in fact, do not line up. You can't make these the same just by rotating things. So they are indeed different. Fructose is also C6H12O6, but because it has this extra bit here, we can see that it is quite a different structure. So it's more obviously different. Now, these monosaccharides, these are these simple sugars that the body can most easily break down. And we're not going to get into the metabolism of that. You're welcome to look that up if that's something you're interested in. What we are going to look at is some other ways that we often see these molecules. So even though it's nice and convenient to draw them in this straight chain form where we can easily see where everything is, in reality, they don't spend a lot of time in this configuration. And so sugars actually link up by doing this kind of a thing. So remember our line structures here. Each one of these things is a carbon atom. So if you imagine this whole thing folding up on itself, what happens is this O ends up connected to this C, and this O over here turns into an OH, and you end up with these ring-type structures. Sometimes it's a 6-membered ring, 1, 2, 3, 4, 5, 6. Sometimes it's a 5-membered ring. And the rings are often drawn with these straight bonds up and down because that whether an OH is up or down affects which sugar it is. So that's the ring equivalent of being on the right or left in that previous structure. So now we have even a couple more forms of this. So if you see things like alpha and beta forms of sugars, you're talking about these ring structures. Now, I'm not expecting you to memorize these structures or to be able to interconvert between something like the straight ones that we saw earlier and these ring ones here. All I want you to know is that these are all forms that you'll find sugars in. And so when you see these types of structures, you should be able to identify them as simple sugars, as monosaccharides. All right. So then moving on from those, we can also have disaccharides. You can probably guess what that means. Monosaccharide was one small sugar unit. Disaccharides are two of those linked together. So if we take a molecule of glucose and a molecule of fructose, and we link them together with something called a glycosidic or glycoside bond, we end up with a disaccharide. Now let's look at what's happening here, and it's color coded here if you can see that, which is helpful. We've got an OH here and an OH here, and notice that this OH and this H are in red. And that's because when they connect, the H and the OH actually disappear. They're gone. This is not a very well drawn chemical reaction because it doesn't account for all the atoms. But what's happening there is we're losing water. Remember, H2O is water. So if we make a connection where H2 and O are not there anymore, then that's water. So this is sometimes known as a dehydration reaction or also a condensation. So dehydration or condensation is what's happening when those link up like that. For kind of like a top view, back to our normal line structures, it would look something like this bottom one. So you have glucose and fructose, and this new disaccharide is called sucrose, and sucrose is table sugar. So that's the regular white sugar that you get. And it's refined down to this molecule. So whether that's corn sugar, beet sugar, cane sugar, if it's white refined sugar, it's just this stuff. It doesn't really matter where it came from in terms of like chemically what it is. All right, so that's a disaccharide. Now, another interesting thing about sucrose here. So here's our sucrose, and you can see it's made of one glucose molecule and one fructose molecule. So when that's broken down in your body, this glycoside bond breaks, and you get some glucose and some fructose, about half and half, and you process those. Another popular sweetener is something called high fructose corn syrup, and high fructose corn syrup is made from corn. But one thing that a lot of people don't know is the fructose content is not actually much higher than it is in sucrose. It's pretty similar. It's right around 50%. The high in high fructose comes from the fact that it's much higher than regular corn syrup, which if you've ever tasted it is much less sweet than sugar. So what they do is they break down the corn syrup a little bit to make a syrup that has more fructose in it and more carefully mimics sugar. There's nothing particularly special about high fructose corn syrup. It's just another sweetener. It's very similar to sucrose chemically. Behaves so in your body too. So some other sugars you may have heard of, like lactose, milk sugar, maltose. Maltose is the sugars that come from germinating seeds that often involved in like fermentation. Things like beer and stuff like that come from maltose. And there are various different disaccharides. Now you can see here, these disaccharides are made in slightly different ways. So for example, sucrose was made of a glucose and a fructose, whereas lactose is made of a glucose and a galactose. And depending on the orientation that these things are in, that makes a slightly different structure for how they connect there. All right, and then we get to our final category of sugars or carbohydrates, polysaccharides, also known as sometimes starches or cellulose. And these are what we call polymers of glucose. So this is where you take a glucose molecule and connect it to itself many, 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 many times. And if you do that, you get various things like amylose and amylopectin, which are the major components of things like rice and potatoes. These are your basic starches. And so you've got this like glucose connected to glucose repeated n times. So many, many, many glucoses repeated over and over again. And then amylopectin, same idea, but then it also kind of branches out, makes this branched structure. So that's going to change the way those things behave a little bit. And that's why, like, for example, long grain rice behaves a little bit differently from short grain rice because of the amylose to amylopectin ratios, and things like cornstarch. The reason that it behaves the way that it does is because of these starches that it has in there. And then the final polysaccharide that we'll look at is cellulose. So this isn't necessarily a great structure. Cellulose is also just a long polymer of glucose, but it is linked up in such a way that the chains can lay on each other and hydrogen bond with each other. So you end up with this very, very strong network, and that's what a plant cell wall is made out of, which gives the plant structural support. So wood and paper are cellulosic in nature, as it says here, leaves, stuff like that. Anything structural, stems on a plant is mostly cellulose. And that's why when you eat things like celery or, lettuces, kale, whatever, there's a lot of fiber in there. That's cellulose that we don't digest. Yeah, you need to have your stomachs or a digestive system of something like a cow to be able to digest cellulose. So we don't get nutrition from it. We can't break it down because of those strong linkages there. All right. So that's basically carbohydrates. So just to review, lots of OHs. They can have a ring structure or a straight structure. And then you can have monosaccharides, simple sugars, or disaccharides. Disaccharides are also kind of simple sugars usually. And then polysaccharides, which sometimes we call complex carbohydrates or starch or cellulose. We're not going to get into Fisher and Hayworth projections in this class. We're just going to leave it at that. All right. So let's jump over to our next category, which is lipids. So as we said, lipids are the non-polar parts of the macronutrients. So where the carbohydrates had lots and lots of OHs, lipids are going to be made mostly of carbon-carbon and carbon-hydrogen bonds. Those are non-polar, kind of like the alkanes from that properties of organic molecules activity last unit. All right, so things like fats and oils. So let's take a look at some of those structures. If we were on campus in lab, we would do a really neat lab about this stuff where we would make biodiesel from vegetable oil. We can't do that this semester. It's not really safe enough to do at home, although people do it at home, but they probably, I don't know. I'm sure some of them take precautions, but probably some of them also don't. So here's how certain types of fats are generally put together. Take a look at this structure right here in the middle. This is a fatty acid. And fatty acids all have similar structure to this. A really, really long carbon chain surrounded by hydrogens, and then a carbon double bond to oxygen with an OH at the end. That's called a carboxylic acid functional group, and that's the end of the fatty acid. That's why it's called a fatty acid. All right, so long carbon chain, and then this thing. Those fatty acids can link to a molecule called glycerol, sometimes called glycerin, same thing, which is just 3 carbons with OHs. It really kind of looks like a carbohydrate almost. And if you notice, kind of like in the last one, we've got the H here highlighted and the OH highlighted here. So if we get rid of water, we get rid of the H, we get rid of the OH, and we instead connect the O to the C, we get a glyceride. And if we do that 3 times, we get a triglyceride. So even though it's all stretched out here, on the left is our glycerol piece that's now connected to fatty acids, and it's connected to 3 of them. So that's what we call a triglyceride, which is generally known as a fat. That's what, when we talk about like a vegetable oil or an animal fat or whatever, you're talking about triglycerides, which are chemically these. You can also just have 1 or 2 fatty acids connected to the glycerol backbone, and then you have a monoglyceride or a diglyceride. So if you see things like that on your ingredients, monoglycerides, diglycerides, those are just basically like fats, but without all the chains. And that usually results in a lowering of the melting point. And we'll get to that in a second. So one thing that we see in some different fatty acids is whether or not they have double bonds. So this one up here, which is stearic acid, you can see there's no double bonds in the carbons at all. And this one, which is oleic acid, a major component of olive oil and vegetable oil, you can see it has a double bond right here. So what that results in is that this saturated, so I'm getting ahead of myself, this fatty acid down here has fewer hydrogens than this one up here. You can see the hydrogens that were here are now missing because carbon has to have 4 bonds, and it gets it from the double bond in this case. So we call any fat that has double bonds in it an unsaturated fatty acid, or an unsaturated fat if it's a triglyceride. And if it doesn't have those, it's saturated fat, right? So that's where those terms come from, if you've probably heard them before. And the reason that happens, or the reason that that makes a difference, is if we again go back to the actual structure of these things. If we were to draw it with the proper angles, we would get something like this, or if you imagine like a line structure. When you put in that double bond, what happens is it causes the chain to turn. It causes this kind of kink in the chain. And so now the fatty acid chains can't line up and have as strong of intermolecular forces with each other. And so because of that, they're not going to have as strong of attractions, and so they're going to melt more easily. So saturated fats are generally more solid at room temperature because of those strong intermolecular forces, those strong dispersion forces. So things like butter, lard, coconut oil, those all have a lot of saturated fats in them, saturated fatty acids, whereas things like vegetable oil, olive oil tend to have unsaturated fatty acids like this. There was also a time where it was popular to use these things called trans fatty acids. So the natural fatty acids like this are generally cis, which means that the H's are on the same side of the double bond. Trans, they're on opposite sides, although I would have drawn it a little bit differently there, but that's okay. And the difference here is these really act more like a saturated fatty acid. There are naturally occurring ones as well. But it used to be popular to put these in snack foods because they have really, really long shelf lives, and they could advertise that the foods didn't have saturated fat in them because technically these are unsaturated fats. Now we know these are actually worse for you than the saturated fats were. So most foods, mostly these aren't used anymore. But if you see something like a partially hydrogenated something or other in your ingredients, that's what this is, a little bit of trans fat in there. Another term you may have heard are things like omega fatty acids, like omega-3 or omega-6 fatty acid. And what that refers to is where the double bond is compared to the end of the chain. So the end of the chain, the very last carbon on a chain away from the carboxylic acid part, is called the omega carbon. So the alpha one is right next to the carboxylic acid, right next to the functional group, and the omega one is the farthest one away. So if you count in from the omega, then that gives you, wherever that double bond is, that's where that term comes from. So this is an omega-3 fatty acid because we go 1, 2, 3. The double bond is on that third carbon in from the omega. So it actually has multiple double bonds in it. So this would be a polyunsaturated fatty acid. And the reason those are important is that they're used in producing other types of lipids. So you need to eat these types of fatty acids, which you get from things like fish, so that your body, and other places, nuts and stuff too, and so then your body can use those to make other types of molecules that you need. All right. There's also waxes. So if you imagine things like, like earwax, the stuff in your nose, things like that are kind of waxy, as well as plant-based waxes like carnauba wax or paraffin or whatever. Those are also lipids. That's what happens when you take fatty acid chains and you link them up with other long chain molecules. You make waxes. Right. A really important category of lipids in the body are the phospholipids. So phospholipids, as you could maybe imagine, is a fatty acid that is linked, or a fat that's linked to phosphorus, or in this case, phosphate, PO4. So you can see the picture here. You have fatty acids made of glycerides. So this would be a diglyceride here with glycerol, and then connected to a phosphate. And that whole thing kind of looks like this if you imagine all of the atoms as balls, and we can actually just represent that as this. And the head of this molecule with the phosphate is very hydrophilic, which means it's really polar. It wants to attach to charged things like water. But the tails are these fatty acids, which we've talked about are very nonpolar because they're mostly made of carbon and hydrogen. So we can look at we can look at this as a molecule that has kind of two different sides, the hydrophilic head and the hydrophobic tail. And these organize themselves in a way that looks kind of like this. So there again are our heads and our tails, our hydrophilic heads and our hydrophobic tails. So hydrophobic meaning nonpolar. This is what cell membranes are made out of. So the outside of the cell membrane is hydrophilic and therefore can interact with the water and, get around in your body. And then the lipid layer prevents polar stuff from getting into the cell that isn't supposed to get in there. And so that's actually is there a picture of it here? That's an important part of drug design when we're trying to make like medications. Something might chemically do exactly the thing you want it to do, but because of the molecule structure, it can't get through that phospholipid bilayer and actually do what it's supposed to do in the cell. So the molecule has to be altered, changed a little bit to try to get that to work. All right. And then the last ones we'll talk about, there are some other categories of lipids as well, but the steroids are an important class too. And again, one way you can recognize all of these is just by looking for lots of carbon and hydrogen. The carbohydrates that we already talked about and the proteins that we're going to talk about don't have as dense of carbon and hydrogen type areas. And so that's a way that you can distinguish them. So even in these totally different looking molecules, these steroids don't look anything like fatty acids really, but they're mostly made of carbon-carbon bonds with carbon hydrogen around the outside. So something like cholesterol, cortisol. If there are some, we can look at, if you look up some hormone structures, anything that ends in an "-one" is probably, like progesterone, testosterone, things like that are steroids as well. And you can recognize them by their very distinct 4-ring structure. So 3 6-membered rings and a 5-membered ring. There's really nothing else that you'll see that has those types of structures. So nonpolar, looking for things that don't have a lot of OH, those would be the lipids, mostly carbon and hydrogen. All right. Let's then go to the proteins. So you've undoubtedly heard of proteins before. Lots of different things that proteins do throughout the body. So for example, here's a table of some different types of proteins in the body and what they do. So we've got digestive proteins, transport proteins, and some of these connect with some of the things we've already talked about. So for example, amylase is a digestive enzyme that breaks down amylose, the starch. So lipase is an enzyme that breaks down fats, lipids, so forth. So those are digestive enzymes. So proteins can be functional, can be things that, can be catalytic. As an enzyme, they help facilitate chemical reactions. They can help transport things around. So like a hemoglobin protein transports oxygen through the blood. They can be structural. So proteins that just give you structure, like keratin, which is what your fingernails are made out of. You can have hormones that are proteins. So some of the hormones are molecules like we saw that would fit more in the steroid or lipid category. But there are also protein-based hormones like insulin. They are involved in defense. Immunoglobulins are proteins that can try to destroy invaders, pathogens and stuff. Contractile proteins like muscles, that's where we often probably most or often commonly associate with proteins, but that's just one type of protein that can contract and change its size. And a bunch of those doing that is what causes muscles to do what they do. And then there's storage proteins that store nourishment for later. So things like in an embryo or in an egg or something like that, those might store some proteins for the developing seed or um, organism or whatever. So lots of different types of proteins. They do lots of important things. All right, so let's kind of go talk about protein structure. When we talk about protein structure, there are 4 different levels of protein structure depending on how far we're zoomed in. So we can look at the individual, like the atomic level, and how the actual atoms are connected. That's called the primary structure. We can zoom out a little bit and kind of see how that overall chain starts to look. That's called the secondary structure. Then we can go out further, and we have what's, called the tertiary structure, and then the quaternary structure further out from that. So we're going to take a look at each of those and what that means and how you can identify proteins at each level. So first, we'll get down into the amino acids. You've probably heard of amino acids before. They are the building blocks of proteins. They're the smallest unit that proteins are made out of. And they're very, very small molecules that look like this. So you have a carbon, double bond oxygen, and then OH. So once again, that's this acid group. It says carboxyl here. That's another way of describing that. We could also say it's a carboxylic acid or just an acid. So much like with the fatty acids, we've got a similar thing going on here, and that's where that acid word comes from, an amino acid. Then on the other side of the amino acid, on the left side, we have a nitrogen, which is called an amino group. That's just the name given to that nitrogen functional group. So that's NH2, and that's where the amino name comes from. So you got amino and you got acid. And then right in the middle, connecting those two, is one more carbon that's called the alpha carbon, and this R group called the side chain. And every amino acid, whatever ones you've heard of, glycine, alanine, tyrosine, whatever, all look like this. The only way they differ is by their side chain. So the different side chain lead to different amino acids, and then the combination of different amino acids is what gives you different proteins. So let's take a look at those things combining together. Well, we can look at a chart of amino acids. You don't have to memorize these. If you take a biochem class or you go into a health science or something, you'll probably have to learn them at some point. Nutrition, dietetics, something like that. But here you don't. But just so we can kind of look at them, some of these names are probably familiar: glycine, alanine, histidine, maybe, I don't know, aspartate, or sometimes called aspartic acid, phenylalanine, tryptophan. I don't know if you've heard these names or not, but these are what these different things are called. And you can see that the side chain for each is highlighted in blue. So having that different thing down there gives it a different function. And in this table, they're categorized into different types of side chains. So you can have non-polar side chains that are mostly carbon and hydrogen, like this. You can have polar ones that have OH, SH, NH, like these. You can have positively charged side chains. So there's little pluses on the nitrogens in these over here, as well as some negatively charged ones. And then you can have these big, what are called aromatic groups, these alternating double bond rings, which make them predominantly nonpolar as well. So those are different side chains. There's one particularly strange amino acid called proline, which is this one here. And proline's different because the nitrogen, the amino part, is actually involved in a ring with the side chain. So it's actually connected to the side chain. None of the other ones do that. And so that means that when proline is in a chain of amino acids, it's actually going to tweak the structure, kind of like those double bonds in the fatty acids did, and cause a different type of structure. And we'll get to that when we talk about the secondary structure next. All right, so 2 amino acids can come together like this, and just like we've seen in the last 2 categories, can make what's called a peptide bond. So you've got H and OH. You get rid of the H and the OH, and that's called water. So that's a dehydration or condensation reaction. And then what's left over is the carbon and the nitrogen, and those are going to connect together and make this type of a bond, which is called a peptide bond. It's also sometimes called an amide bond, but in the context of proteins, it's usually called a peptide bond. All right. So in this case, this is glycine and glycine. So you'd have two glycines linked together. And this is the basic reaction for how all proteins are then linked together. Okay. So that link that they're going to do here is called the primary structure. And we can see what that looks like here. All right, while that's loading, I'm just going to pull up this other one. There's another book here as well. So here's kind of what happens with the primary protein structure. You've got all these little colored balls here are different amino acids. And as they connect together, we can do what's called a sequence, and we can say which amino acid is connected to which and in which order. And that's the primary structure right there. All right, so you hook these things together, and you get these chains. Sorry. All right, so here's another example of that. Now, these different letters, each amino acid has a one-letter abbreviation. You can look that up. Again, you don't have to memorize that. But by knowing the sequence of letters, we can know that the primary structure of that protein. How are all the amino acids connected together from one end to the other? And so that's the primary structure. So now moving into the secondary structure, what happens is that the shape of that chain sorry. The shape of that chain ends up changing depending on what types of things are connected to each other. So sorry, computer's yelling at me that I've got too many things going on here. Let's see if I can close some of this stuff out here. Bring it back. All right. Well, we may have to stop a little early. We'll see what happens here. I can close my lipids here. Maybe that helps. Well, while we're waiting on that, the secondary structure, there are two major parts of the secondary structure, alpha helix and beta sheet. So as these things start connecting together, you get this. Alpha helix, beta pleated sheet. So either the chain ends up kind of making this sheet that can hydrogen bond with another sheet, or it ends up making this this twisty form, which is called an alpha helix. And it's generally those prolines that cause this thing to twist around like that. That's called the secondary structure. So we can look at, as we're zooming out, we're no longer focusing on which amino acid is connected to which. We're thinking more about what does the structure look like if we kind of zoom out more. And that's the secondary structure. So then, if we zoom out even more, we can think about how these various helices and sheets are all organized together. Because they can actually connect to other ones in a totally different spot on the polymer or on the protein chain, on the amino acid chain. So for example, one of those positive side chain amino acids could find one of the negative side chain amino acids somewhere, and then those two things could end up getting close together and attracting each other. That can happen with hydrogen bonds, that can happen with sulfur, that can happen with nonpolar stuff. So as this thing is twisting around, it can organize itself. If you've ever heard about protein folding, this is what that's all about. That's this tertiary structure. How does this big thing now fold onto itself to make the protein? And the folding is just as important to the function as the individual amino acid lineup. So that's why it's an important problem, and it's actually very difficult to predict how these things are going to form because they are so big. So that's a big area of research, is trying to predict and understand protein folding. And that gives the protein its final three-dimensional shape, right? So those are really the three, that's primary, secondary, and tertiary structure of proteins. Then the final piece is the quaternary structure, where you can have multiple amino acid chains come together to be part of a protein, right? So just to review, we've got amino acids linking up in a particular order to make the primary structure, then forming sheets and helix type structures to make a secondary structure. Then those things fold onto themselves in 3 dimensions to make a tertiary structure. And then multiples of those could come together and make the quaternary structure. And that's like the actual, like the final protein, the way that it would be in your body or wherever. All right, so what do you need to know about this stuff? Well, for proteins, you should know first how to identify an amino acid. Like, what does it look like? What kind of functional groups you're going to look for? And you're going to look for the nitrogen on the one side, the C double bond O, and then OH on the other side, and then the alpha carbon with the side chain in the middle. So that's how you identify an amino acid. Then the amino acids link together to form the protein. And if we're just talking about the sequence of amino acids, that's the primary structure. If we're talking about the helix and the sheet, that's the secondary structure. If we're talking about how it all folds onto itself, that's the tertiary structure. And if we're talking about how multiple chains come together, that's the quaternary structure, the 4th level. All right. So by now, you should have a decent sense of what these molecules look like. You should be able to categorize things as a carbohydrate, a lipid, or a protein. And then within those categories, the ones that we've talked about. So monosaccharide, disaccharide, polysaccharide. The lipids you should be able to categorize into saturated and unsaturated fatty acids, glycerides, steroids, phospholipids. And the proteins we just went over. So let me know if you have any questions about those types of things. You'll get a chance to practice that on the quiz later this week. So thanks, everybody. And let me know if you have any questions. I'll check again, but I don't see anything. So let me know if anything comes up. Otherwise, I'll see you Friday morning for our normal recap and review.